And the first is one of the ECR reps, and this is Andrew Hanton from the Rosalind Institute. Andrew, you know how long you've got?
4 minutes. Will be quicker. Thank you very much. So my name's Andrew. I'm a PhD student at the Rosalind Institute in the lab group of Christine Tate-Burkhardt, and I'm going to briefly introduce some of the content of my poster focused on developing a porcine RNA interference library and tell you a bit about how I've used that to study coronavirus host pathogen interactions. So for those who aren't familiar with RNAi screening, it works by the arrayed silencing of thousands of genes to study phenotypes. And it relies on libraries of siRNAs which are commercially available for a small number of model species, including human and mouse. However, there are no commercially available libraries of siRNAs for non-model animals, including livestock and companion animals. So the consequence of this is that the application of RNAi screening in these species has been significantly limited, including in studying their pathogens, including the many important livestock and companion animal coronaviruses. So the focus of my PhD has been to develop a porcine siRNA library and apply that to screening for porcine coronavirus host factors. So after we designed the library, we applied this to screening host factors involved in transmissible gastroenteritis virus replication. We began with the transfection of our custom library into swine testis cells, and after allowing for gene knockdown, we infected these cells with TGEV at a low MOI for multiple replication cycles. And after 48 hours, we fixed our cells, we stained for the N protein, and we quantified the immunofluorescent signal by plate reader. Now, importantly, our screen is the first use of siRNA screening in a livestock model and the first use of siRNA screening to study coronavirus host factors in a non-human model. So being able to study replication of non-human coronaviruses in native species models gives us the ability to generate novel insights, learn more about how these viruses replicate and interact with their native host. So the screen itself demonstrated reliable repeatability, and by implementing a logistic spatial normalization method, we were able to reliably account for spatial variation and things like that. Additionally, our controls that we used in the screen performed reliably, including the host receptor and PEP. And by our method, we were able to identify 250 antiviral hits and 294 proviral hits, including the already described TGEV host factors DIRC1A and HF1 alpha, which makes us reassured that our screen reflects proper host factors. So by implementing a string pathway analysis, we were able to identify many different host pathways involved in our proviral hits, including pathways including vesicular and endosomal transport, important for viral entry, cytoskeletal proteins, many of them, and proteins involved in signaling pathways, including Rho-GTPase and Wnt signaling. So if you're interested in that, and if you want to learn more about how we developed the library, how we conducted the screen, and what our data represents, please come and visit me at my poster number 7, and I'd be very happy to speak more about it. Thank you very much.